video and audio now because I told now I'm joining. Yeah, it is
Yeah.
streaming live on the YouTube. Good.
Okay.
It
Okay,
is.
you can
Yeah.
see.
Yes. Hi, Rupashi ma'am. Hi, Manmohan sir. Ankita is there.
Um,
Hello, ma'am.
Yeah.
Good afternoon,
Yeah.
all.
Good afternoon,
Good
ma'am.
afternoon, sir.
Yeah.
I'll do one thing. I will copy the stream link and put it in the chat box.
Yes.
Yes, ma'am.
And then Ankita can take care from there. Na,
Yeah.
Ankita? Yeah.
Yeah.
Yes, ma'am. Yes, ma'am.
Yeah.
So officially the program starts at 2:30, and I think all the students group know that.
Yeah. So maybe from the chat box, it could be
She can
copied
copy it.
to
Yeah.
the
Yes, ma'am.
to
I'm
the
copying.
Yeah.
Yeah.
yeah
Just
to the
that we
WhatsApp
will take
groups.
care. That we will take care, Mrinalini. Okay, to send it to the students. Okay. Now we have to take care of the recording as well. You just check if you can record.
Yeah, recording I will be able to do.
Yeah. That was the only thing. Your audio and video is clear. My audio and video. Saujanya, have you joined? Saujanya Nagendra? Anybody from Chaitra Jarang? Chaitra?
So Priyanka,
Yeah.
what I will do is as each one speaks,
Hmm.
I will spotlight them, and then after we finish our slot,
Hmm.
like if somebody is introducing, they finish their slot, it's better they switch off their video. That will
Yeah.
be the best way.
Yes.
So
Yes.
that yeah, we will
Only while
let's have an
speaking we will keep
understanding
it
on
on.
that.
Yeah.
Yeah, let's
Only
have
while
an
keep we
understanding on
will.
that. Yeah.
Yeah.
Yeah. So when the speaker is speaking, I think it's better all of us will switch off the video.
Yeah. Yes. That is there. And is Aruna madam there? No, Dr. Aruna is not there. First semester students, it is for the first time for them. So somebody has to guide them. Few of them can join here and a few of them can see on the YouTube because they don't know anything about the, yeah. So can we give this? Yeah.
Students.
Third
Hmm.
semester IBS can take care of that, I think. Students,
Yeah.
they are your juniors. Spandana.
Students, you could, if you are, if we have time and you're allowed, then maybe you can unmute and ask the question. Otherwise, please put your questions in the chat box
Hmm.
and as well as in the YouTube. Okay, so that will be taken care of. So just to avoid any last minute chaos.
So I will ask a chat box question, Mrinalini. I am going to read out, and
Mm,
YouTube questions I
yeah,
will
yeah,
ask you to
yeah.
read at the end. Okay. I'll ask you if there is any YouTube question, then you can ask. Okay.
No, that's okay. That even you can read out. But then I was just telling because if
YouTube,
somebody
you'll be no. Why I'm
unmutes and then
asking no? You can see the
yeah,
YouTube
because
now. I
YouTube
won't be
is
going to
yeah,
YouTube.
yeah, yeah,
Yeah, that's why.
yeah,
YouTube questions, you take care. Chat box question, I'll
yeah,
take care. Okay.
yeah.
Okay. So how many we have? 35. 2:15. We have told the students to join,
Okay.
and you have to let in Saujanya Nagendra in, Mrinalini.
Oh yeah,
She is the
yeah,
president.
yeah.
And that's why I asked that headache of getting the students in, no? Who we have to, because you have to stream here. We have around 170 students now, na? No, 150.
Anjali ma'am,
She
Anjali
joined.
ma'am is here.
No, she's not there. Is she there? No. Anil sir is there. Hello, Anil sir.
Hello, ma'am.
Yeah, she's still not logged in. Yeah, yeah. 150. No, she's not yet joined. Is the brightness on my end enough? I have to be a little brighter.
Yes, ma'am, it is fine now. It's fine.
No, because the sunlight is.
Hmm.
Somebody is speaking over the phone. Kindly
ಮೇಡಮ್
mute.
ನಿಮ್ ಬ್ರೈಟ್ನೆಸ್ ಜಾಸ್ತಿ ಇದೆ.
ಜಾಸ್ತಿ ಆಯ್ತಲ್ಲ?
ಯೂಟ್ಯೂಬ್ ಅಲ್ಲಿ ಬ್ರೈಟ್ನೆಸ್ ಜಾಸ್ತಿ ಇದೆ.
ಹೌದಾ? It's fine now. Hello, ma'am. Yeah, one sec. Ma'am, 1 second. Oh, yeah, I'm admitting. Yeah.
Anil sir, you see even my brightness, no? How? Because I'm sitting at a new place today. Is it fine? Yeah, Mrinali,
You are
can.
looking little dull today.
No, this is my. I'm doing
Dark
it.
behind it is dark.
Behind it is dark.
Mm.
Okay, I'll just check if I can do anything about it. Wait.
1 second once.
Yes, madam.
My brightness, no? How?
Mrinalini? Yeah, Priyanka ma'am.
Yes, ma'am. Hi, ma'am. Ankita became the host. Mrinali dropped
Ma'am,
off.
I'm the host.
Yeah.
Yeah.
Okay. Yeah, that's very nice, Ankita, Mrinalini, to put on YouTube.
And the lag is also very little, it seems, na? Lag is almost nahi hai abhi. Ankita joined back, I think. Mrinali joined back.
Mrinalini,
Yeah.
ma'am.
Ankita, the speaker will join at what time?
2:25 or 2:20, ma'am.
Okay. Okay, and there is an announcement for the students. The portal of fees has been opened and you can pay the fees on or before 19 December without fine. Okay. The students, there is an announcement for you because the talk has not yet formally started. The students who have not yet paid the fees can pay it by 19th of December without any fine as per our chairman's notification. Okay.
Saujanya is here.
Saujanya, can you please switch on your video? She's joining. Yeah, you don't see her. Saujanya Nagendra, 9th semester.
Yes, ma'am.
Yeah. Can I just see your video, please?
Yes.
Yeah, good.
Yeah.
So your music box is ready, Nama?
Yes, ma'am.
Yeah.
Ankita,
Yes, ma'am.
ನನ್ಗೆ one clarification ಬೇಕಿತ್ತು.
ಹಾ.
In my YouTube, if I pause, will it be
Hmm.
paused for everybody?
Yes.
Yes, ma'am.
Yes.
Okay.
So you have to be live throughout the program.
Once you can pause and see now itself.
Yeah.
That's what I did it. I did
It
that.
paused. It paused. It paused.
Yeah.
And
that's
it
what.
paused. And actually, when you paused, your this also. What is it? You went out of the meeting as well.
Ma'am, you can give permission to others for recording. Is it possible?
Let me just check.
ಒಂದು ಗಂಟೆ
Meeting is
Class
not
one.
getting recorded, no?
Yes.
Yeah, it will have to be recorded. Venkatesh sir, kindly
ಹಾ
mute.
ನಮಸ್ತೆ ಮೇಡಮ್
Namaste sir.
Kindly mute ಮಾಡಿ sir.
Mute.
ಓಕೆ ಮ್ಯೂಟ್
Okay, Ankita.
Ma'am, you can make Priyanka ma'am host.
Then YouTube will stop.
Oh no. Record
YouTube.
permission. Recording permission. Is it possible?
Recording, I'll have to just press that button. That's all right.
No,
ठीक है.
ma'am.
Recording can be done. Because in case it stops, that's my concern. Otherwise, anybody can be made the host.
Yes.
Yeah.
No.
Yes,
You cannot make anybody else the host. You only have to be the host for the YouTube live thing because it is connected to your account, I think.
Yeah. How about sir?
So unfortunately, it seems sir is busy in some task, and he will try to join, and he may not be available. So we all faculty have to take care of this inaugural ceremony if sir doesn't join.
Anjali ma'am has so many trophies in her background.
You
Yeah.
don't know the real one. Virtual.
No, madam, we can see real or virtual
Whatever we
trophies are
can
trophies.
This one corner
Are
at
they-
least. Kids.
Kids. Not you.
No, not me.
Students, we have only 57 participants, including the teachers. I would like, please ask your friends to join here. YouTube is the second option. First, you have to try here, and we should have that 100 number. Then only you go and see it live on YouTube. Okay? We can see 60, 61, it's increasing.
Priyanka, he's
Yes.
joining. The speaker is joining.
Okay.
Is it Govindraju T?
Yes.
Yes.
Govindraju T.
Okay, yeah.
Hello, Dr. Govindraju.
Hi.
Welcome. Welcome
Is
to the
it Ankita?
department. Yeah, Ankita is there.
Okay. Hi. Hello.
Good afternoon, sir.
Good afternoon. Good afternoon to all of you.
So thank you, sir, for accepting our invitation.
Yeah, it was too short notice,
Yeah.
but because Ganesh is good friend of mine, and more than that, Bangalore University is my alma mater, so I couldn't say no.
Okay, sir.
So,
Okay, so just one thing. Can you just give the title of the talk? I think.
yeah. So I'll share the slide then.
So not you can share. You can just test it because we have this is
Okay.
the inaugural ceremony. So we have a small Ganesh Vandana and Admele. That what is this ceremony about? A small introduction of
Okay.
that. Then your
Okay.
talk will start. So
Okay.
you
I'll
can
just
test
share
it
the
now.
slide so that you can also note down
Yes. Sure.
the title.
Yeah, we can test it also. Yeah. Thank you, sir.
Can you see the slide now?
Yes, it's visible. It's visible. Perfect.
Yeah.
Alzheimer's multifactorial disease.
Alzheimer's is a multifactorial disease. Yeah. Hope that suits
Interesting
the
topic.
purpose.
Yeah.
Yeah.
It serves.
Okay.
Interesting topic. Thank you.
Okay. So I'll come out. I'll unshare. Right.
Priyanka, inform sir that we are going live.
Yes.
Yeah, just a minute. I'm trying to unshare it.
Sir, on the
Oh,
top,
right here.
top
Yeah,
panel.
I
Yeah.
got it.
Yeah.
Yeah. Thank you.
I'll inform her. Sir, maybe our chairman actually has a very important urgent meeting, which was also a last minute. So he will try to join. He has conveyed that to you, and he has thanked you also.
No problem. No problem.
Okay, sir. So
Yeah.
we have all the faculties present here of the
Sure,
department.
sure.
Okay, sir.
Absolutely.
And our
Yeah.
students. Yeah.
Yeah.
Just we'll in 5 minutes we'll start. Ankita, ನೀವು ಇನ್ಫಾರ್ಮ್ ಮಾಡ್ತೀರಾ ಸರ್ಗೆ that we are going live and we are starting?
Ma'am, you can only inform. Yeah.
Sir? Okay.
Yeah.
Okay. Because I messaged him, it has not reached him. I think we can. Anjali, madam, should we start? Because sir has, I informed, sir has not seen the message.
Yeah, ma'am.
Huh? Okay.
I will just call him otherwise. I'll just call him.
Yeah, ma'am. Just call him once. Thank you. Thank you so much.
Hello, Dr. Govind Raju. I'm Dr. Mrinalini here. We are
Hello,
going.
yeah.
We are going on YouTube Live.
Sure.
You could also share the link for anybody else who would be interested in listening to your talk.
Yeah, sure. I don't think I will be sharing now. So whomever you are
Okay.
sharing, I think that's it. Yeah. So
Okay, sir. Thank you.
yeah.
No, he is not reachable. Can you try it out, Ankita?
Yes, ma'am. I'm trying.
Just 2 minutes of this technical glitch and then we will go ahead.
Maybe let me ask you, one of you can probably tell me who are the audience?
So
Sir, audience is BSc, MSc integrated course we have.
Okay.
We have MSc Life Science course.
Sure. Okay.
A couple of research scholars also from the department.
Okay.
So actually we had got this opportunity to listen to your talk in the peptide conference at Bangalore Central University, I think last year.
Oh, okay. Okay. Yeah, there I was kind of too many things. More of a, because yeah. So today will be more focus on one subject.
Yes, sir.
Yeah. Yeah, hope I will take you all, the mix audience. I mean, mix audience in different level. So I'll try to take them along. Sure. Thank you.
Sure. Thank you.
I think I'll start the recording now. It's 2:30.
Yes.
Ma'am, I called sir and he said we can start the program.
Okay.
And he will join
Okay.
like after a few minutes.
Okay.
Okay.
So,
Ankita, what do you suggest I record to the cloud or on the computer?
Better, ma'am, computer.
Okay.
Yeah.
okay. So a very warm greeting to our honorable guest speaker, Professor T. Govinda Raju. My dear colleagues and the enthusiastic audience present today on this online forum. On the behalf of the Department of Life Science, Bangalore University, I welcome you all to the inauguration ceremony of the BioRam Forum activities for the academic year 2020 to 2021. The year 2020 has been unforgettable due to a zillion of reasons for all of us. Due to the COVID-19 pandemic, the world came to a standstill. But the resilience of humankind against all odds proved that there was never a night that could defeat the sunrise. As a result, social distancing, virtual interactions, and the online life became the new normal. The BioRam Forum of the Department of Life Science organized many online talks and webinars under its umbrella in the past academic year. As most of us know, every year the BioRam Forum provides our students ample opportunities for extra and co-curricular activities. Today, we are here to celebrate the inauguration of the BioRam Forum activities for the current academic year. We are very honored to have with us Professor T. Govindaraju from the Bioorganic Chemistry Laboratory of the esteemed Jawaharlal Nehru Center for Advanced Scientific Research, JNCSR. He will enlighten us today with his talk to mark the inauguration of the BioRam Forum activities for our department. And before we start, it is the custom of our department to mark every new beginning with the invocation of the Almighty. Vakratunda Mahakaya Suryakoti Samaprabha Nirvignam Kurume Deva Sarvakaryeshu Sarvada. We have with us Saujanya Nagendra from 9th semester of Integrated Biological Sciences, who will present Ganesh Vandana before us today. Saujanya, over to you.
Yes. Am I audible?
Yes.
You're audible. Yes.
Okay. Gam Ganapate Namo Namo Shankari Tanaya Namo Namo Gam Ganapate Namo Namo Shankari Tanaya Namo Namo Gam Ganapate Namo Namo Ankushadhara Mangalakara Pankajanayana Ankushadhara Mangalakara Pankajanayana Ankushadhara Mangalakara Pankajanayana Varana Sariga Gari Sani Pani Sari Risani Paga Pani Sa Sani Pagari Sari Gare Ga Paga Pani Pani Sani Sari Sari Gari Sani Pari Sani Paga Sani Paga Ni Pagari Sa Sari Ga Pani Sa Sari Ga Pani Sa Sari Ga Pani Sa Gam Ganapate Namo Namo Shankari Tanaya Namo Namo Gam Ganapate Namo Namo Pankajasana Vandita Bhaktarti Harana Mudita Kumkumambu Gunanidhi Hari Kesha Kumara Kumkumambu Gunanidhi Hari Kesha Kumara Mandaragam Ganapate Namo Namo Shankari Tanaya Namo Namo Gam Ganapate Namo Namo Thank you.
Thank you so much for that beautiful rendition, Saujanya. Now, I would like to request Dr. Mrinalini BR for giving us a brief introduction about the BioRam Forum of our department. Over to you, Dr. Mrinalini.
Thank you, Dr. Priyanka. A very good afternoon to the august audience here. I take this pleasure to introduce the BioRam Forum to all of you. BioRam Forum is a seminar club, the brainchild of Professor Puttaraju, ex-chairman, Department of Life Science. The forum was initiated in 2008, and it provides a platform for students of integrated BSc, MSc Biological Sciences and MSc Life Science to present seminars on a topic related to their syllabus each semester. The forum also encourages invited talks from eminent scientists or experts in the field of life science so as to update the students with current research activities and scope for future research. The forum's activities are extended to conducting study tours to various institutes, industries, museums, sanctuaries, reserve forests, etc., to corroborate their classroom learning with recent developments. The BioRam Forum also regularly conducts sports and cultural events adjudged by resource personnel drawing from respective fields. Maximum emphasis is given to the overall development of students in cultural activities, sports in parallel to academic achievements. It is very satisfactory to note that in spite of the COVID situation, we at the Department of Life Science, under the leadership of our chairman, Professor Shivashankar, conducted a week-long webinar series on Zoom platform in the month of August as part of the BioRam Forum activities. Eminent speakers from across the country gave talks on topics of relevance and importance to the present situation. Today's talk is hence a part of the BioRam Forum activity, and I request the students and faculty to make the best use of it. Thank you.
Thank you so much, Dr. Mrinalini, for that nice, lovely introduction of our BioRam Forum. And I would now like to request Dr. Anjali Ji to introduce our esteemed guest, Professor T. Govindaraju, to the audience and invite him for the talk. Over to you, Anjali, madam.
Thank you, Dr. Priyanka. Good afternoon, one and all. It gives me immense pleasure to introduce our distinguished speaker for today's webinar, Dr. T. Govindaraju, Professor, Bioorganic Chemistry Laboratory, Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore. Dr. Govindaraju's path-breaking research has led to the discovery of natural product-based therapeutic candidates for Alzheimer's disease. The group has modified the structure of berberine, a natural and a cheap product similar to curcumin, available commercially into Bird-D to use as an Alzheimer's inhibitor. Their research work has been published in the scientific journal iScience. Working at the interface of chemistry, biology, and biomaterial science, his group has developed an injectable silk fibroin-based hydrogel for sustained insulin delivery in diabetic patients. Dear students, you will be amazed to know that Dr. Govindaraju has done his master's from Bangalore University. He pursued his PhD from National Chemical Laboratory, Pune, under the guidance of Professor K. N. Ganesh. He completed his postdoctoral research from University of Wisconsin-Madison, USA. He has been selected as a Humboldt Foundation Research Fellow at Max Planck Institute of Molecular Physiology, Germany. He has been recently awarded National Prize for Research on Chemistry of Peptides and Nucleic Acids, CNR Rao Education Foundation. He serves as a Secretary, Indian Peptide Society. We are very proud, happy, and feel privileged that he readily agrees to deliver the talk today. With this short introduction, I would now like to invite Dr. Govindaraju for the inaugural talk. Thank you.
So thank you very much for that kind introduction. Of course, let me also thank the Department of Life Sciences and all the faculty, for inviting me, to give this talk. And of course, Ankita was in touch with me. But definitely, I'll thank the department and all the faculty colleagues at Department of Life Science for the opportunity to be giving this talk and especially sharing some of our work with the students of the department. Let me share my slides. Okay, I hope my slides are visible.
Yes, sir. It is visible completely.
Okay, so thank you again. So I'm going to introduce to you Alzheimer's disease from the perspective of the multifactorial disease that the disease is. So I'm going to give you my own perspective, or rather, I would say our perspective on Alzheimer's disease, and why, and also, of course, I will also refer to literature and also from our own research. I'm going to tell you why so difficult, this to tackle this Alzheimer's disease, both from the viewpoint of developing diagnostic and therapeutics, and that's where I will emphasize and try to kind of put across to you, Alzheimer's disease is not a simple disease, rather a complicated disease. Before I do so, let me, since there are a large number of students are participating, so let me briefly tell you, about our research activities at Jawaharlal Nehru Center. So as indicated, am I audible? Clear to you?
Yes, sir.
Yes, sir.
Yes,
Wonderful.
sir, you're
Okay. Yeah. So thank you.
Yes.
So we work at JNCASR, our research group work in the area of bioorganic chemistry and chemical biology. We all know that proteins are the real workhorses in cell. Okay. So well, we have so many biomolecules, but especially proteins. I always, usually I also give, quite a number of talks to school children, and for them I tell them, while nucleic acids, or a DNA is like a king. If you keep all the information and give orders, the proteins are like, kind of a common citizen like you and me. Okay. And we are the ones who do all the work. Okay. So that's how the proteins does all the work in a cell. And of course, there are proteins can be good, and at times proteins can also be bad. So in our lab, we are working on various aspects which covers both these. Our research teams actually, we work on two research teams which covers both these aspects. Meaning, what happens, when the proteins actually, with reference to materials, what happens if proteins are good, material, and of course, what happens when they become, bad, when they become toxic. So those are the themes we work on. Precisely, that's what, when a protein which are useful, beneficial protein based material which are beneficial are called functional amyloids. The best example is silk. Silk is one of the well-known functional amyloid. Of course, I will not go into the detail. That can be another, topic, another talk by itself. So silk, in our lab, we are trying to use silk and its mimetics, for biomaterial applications, including neurogeneration. Of course, we have done quite a bit of very interesting work in this area, and more importantly, it is very close to my heart. So to us, because we all know that this, by using silk, for any other application other than textiles is like a value addition in sericulture, especially for for instance farmers. So from that perspective, so it's definitely very interesting and also very close to my heart. And nevertheless, from the scientific, our own thematic aspect as well. So it is very interesting that there are several functional amyloids, and silk, especially silk fibrin, is one of the well-known and abundant protein-based materials which has innumerable applications. Of course, we are also trying to develop silk mimetics through reductionistic approach. Probably that will be too too much. So again, that is also something new area which is established exclusively in our laboratory, which is also been accepted worldwide. So I will not talk about both of these topics, and hopefully I will have another opportunity to talk about interesting aspects of silk and its mimetics for biomaterial applications. Of course, there are proteins which undergo aggregation. We'll come to know that later in a while. So they undergo aggregation and become toxic, especially in the brain, and which is responsible for many diseases, which we actually call them neurodegenerative diseases. And we in our lab particularly interested in Alzheimer's disease, which is one of the major neurodegenerative disease, to develop diagnostics and therapeutics. Of course, today in this talk, I shall probably try to brief you and try to present to you, or I'll try to say some various aspects of Alzheimer's disease from the viewpoint of both diagnostic and therapeutics, and from our own and especially considering contributions that our lab has done. Well, we all know that proteins are produced on ribosomes in the form of polypeptides. So, which is, I mean, for this audience, it's probably very preliminary slides. So, nevertheless, the proteins are produced on ribosome in the form of nascent polypeptides, which are subjected to protein folding, so that they acquire the three-dimensional structure and become functional. Well, this is close to works close to perfection, but it is not 100% perfect. So at times these polypeptides or proteins, partially folded proteins, can escape this folding process, and then they know what we call misfolded. And of course, in every cell has its own repair mechanism. What it does is it put back this misfolded protein back to refolding mechanism. And if that fails again, then those proteins will be degraded, or digested through proteasomal degradation or autophagy or some other means. But under some adverse physiological conditions, these misfolded proteins, they escape both repair mechanism and degradation mechanism, and then become amyloidogenic. What do I mean by amyloidogenic? Meaning these misfolded proteins undergo ordered aggregation to produce such a highly toxic materials, and which is responsible for at least 25 different neurodegenerative diseases. And Alzheimer's disease is one such disease, in fact, a major disease, which constitute around 70 to 80% of all dementia. So Alzheimer's disease was reported by a German doctor in the same name, almost more than a century ago, while treating this patient. This patient was Augusta Dieter. She was, 56-year-old, and she dies in the within a year. And then postmortem analysis, of course, not just this patient or any Alzheimer's disease patient, is characterized by two kinds of protein aggregates. One is plaques, other one is tangles. I'll come to that. What are these plaques and tangles? Well, although the disease has been reported more than 100 years ago, still there are no diagnoses, no cure. Let me tell you, there is a steep rise in Asia and particularly India. In fact, while all the major diseases, whether it can be heart disease or a cancer, stroke, or AIDS, showing decline in number of deaths. When I say decline, so it's decline because there are it's due to advancement in biomedical research and treatment. So Alzheimer's disease, actually, rose by 71%. Trust me, this is actually 2013 data, and current data, it's actually more than 85% increase in Alzheimer's disease. And in fact, Times Magazine and all over the world, experts, everybody project that Alzheimer's disease, is going to be #1 disease very soon. Very soon, all over the globe. Okay, so that is the impact. So this is going to impact, both public health and economy. Coming to molecular mechanism, there is a transmembrane protein called human amyloid precursor protein, APP, which can be cleaved or processed by a family of proteases. We call them secretases. There is alpha, beta, and gamma. Especially processing of this transmembrane protein, by beta and gamma secretase generate these peptide fragments. We call them amyloid beta or A beta peptides, which are sticky, meaning they undergo aggregation to form polymorphic species, polymorphic aggregation species such as oligomers, protofibrils, and fibrils. Okay, and this process can be, will be, is accelerated by the presence of redox metals such as copper and iron, and not only accelerate the process, but these copper and iron, these are redox metals. and also I'm mentioning only two, but there are also non-biometals like aluminum is also implicated in this process. But nevertheless, I'm only considering its redox metals. So they also form an inclusion complex, and by doing so, they become kind of a factory or a depot for generating reactive oxygen species. These inclusion species will start kind of generating reactive oxygen species, unstoppable production of reactive oxygen species, and that leads to A beta burden, what we call A beta burden, and which is related to oxidative stress and inflammation. And this A beta burden through routes which are not very well understood leads to collapse of microtubule, and in the process the tau, a protein called microtubule binding protein called tau also collapses, and that undergo aggregation to form what we call neurofibrillary triangles. You remember I mentioned about plaques and tangles. These A beta fibrils are are called plaques, and then these neurofibrillary triangles of course are simply called tangles. So these are the two protein aggregates which are characteristic or hallmark of Alzheimer's disease. And now hope you can now understand that Alzheimer that leads to multiple toxicities. So Alzheimer's disease is not a simple linear disease, rather a multifactorial disease. Again, just to recap, if you ask the question to yourself or to me, why there are no diagnosis or drugs for Alzheimer's disease? Well, because Alzheimer's disease is a multifactorial disease characterized by multifaceted toxicity, which involve protein aggregation, which I already mentioned. And then, in fact, at everybody, even the healthy, people, humans, in the brain, they have, some kind of protein aggregates, but there are mechanisms to remove them, clearing. I already mentioned, proteasomal degradation or autophagy or some other route. For some reason, that is, will be, those mechanisms will be switched off or, suppressed in case of Alzheimer's disease. Of course, so you have protein aggregation and malfunctioning in clearance of such, aggregates. And these aggregation species, especially oligomers, also bring in what is called membrane toxicity, very similar to antibacterial kind of bacterial kind of action. And as I already mentioned, generation of reactive oxygen species related oxidative stress and inflammation also plays a major role. I will come back to this. And there are synaptic toxicity, mitochondrial dysfunction, molecule damage, and there are even telomere inhibition of telomerase activity. These are some of the major toxicities, and there are a few others which I have not mentioned. Because of this multifaceted toxicity or a multiple toxicity, the traditional way of drug discovery or even for a diagnostic targeting single biomarker or a target no longer valid or going to work. So for that, we need to go for multi-pronged strategies for both diagnostic and therapeutics. In fact, this is the theme we are pursuing in our lab, in our lab exclusively. So that's how, if you ask how we are, there are many people are working all over the world on Alzheimer's disease. So how we are different. So this is how we are different. So we are not just targeting individual disease pathways or routes, rather we are trying to target multiple disease routes or pathways. Again, I would like to emphasize that Alzheimer's is a true disease, not an inevitable part of aging. I'm sure you will agree with me. This is particularly true. This slide is especially important when I give a talk in India, especially. So I think my research career in JNC is as old as your Bioram Argana Bioram platform. Okay, ours we also started, our research group started in 2008. So when I started in the beginning, whenever I give a talk or talk about Alzheimer's disease, people used to tell me, why Alzheimer's disease? Because it was conceived that Alzheimer's is a disease of Western countries. So unfortunately, fortunately, for for whatever reason, so no longer I get that questions now, because it's very common. It's very well known. Alzheimer's is also spreading. Kind of become a major disease. Even in developing world, and also close to the countries like India, which are also developing and also considering our population. Unfortunately, the awareness, mental health awareness is very, very poor in India. Though that's where it comes. For example, if you go to countryside, or why you go to countryside, even in cities, if somebody become mentally ill, we try to link them for aging. Okay, so on voice I thought, so it's like it's done. So it's something to do with aging. Well, let me tell you, they are two different things. Okay, if you look, if you consider a memory deficit, so normal aging, you see that green trace, you see that is a normal aging. So the memory will never fall to zero. But when it comes to Alzheimer disease, usually, as I mentioned, there is no proper diagnostic for Alzheimer disease. The current diagnosis of Alzheimer disease is mainly on behavioral symptoms or a cognitive decline that usually happens somewhere around this, somewhere 55, 60 years. And from the point, due to visible symptoms, and after that, the memory actually takes a free fall. Okay, I hope it clear, make it, make it, it's clear to you that, so indeed, Alzheimer disease is very, very different, and it is a, true disease, is not inevitable part of aging. And in fact, so this is where I mentioned the current diagnostic happen because of the visible symptoms. But now we know that the symptoms actually generate, start somewhere, probably decades, probably 1 to 2 decades earlier than the current diagnosis. If at all, if you want to interfere both in terms of diagnosis or therapeutic development, so we need to work somewhere here. Further, so National Institute on Aging and Alzheimer Association, they formed a research framework a few years ago. And in 2018, they came up with a research, kind of a set of, a set of rules, okay, are designated biomarkers. They designated these three, A beta, amyloid beta, the plaques I mentioned, tau, neurofibrillary tangles, and related neurodegeneration. Because of these plaques, the neuronal cell die. So that is what we call neurodegeneration. So these must be considered as a designated, these framework designated these three markers as a definite biomarkers. And in fact, they urge the clinicians or doctors to use these biomarkers for a diagnosis of Alzheimer's disease instead of just going by behavioral or a cognitive, functions. Well, interestingly, this framework also left this definite biomarker list of these definite biomarkers open-ended, meaning as and when if somebody actually comes up with a new biomarker, identify and validate a new biomarker that can be added to these definite list of biomarkers. I'll come back to that later on. So coming to diagnostics, okay, so we a few years ago we developed a very interesting near infrared probe which is capable of not only detecting Alzheimer's disease but also distinguish Alzheimer's disease from other neurodegenerative diseases. This is one of its kind, very, very unique. Okay, for example, this is the data from a from a human AD brain sample. So you can see that this is the probe. The sample is co-stained with our probe and also an antibody for tau tangles. So here we consider A beta as a prime biomarker, not the tangles, because the A beta is very specific to Alzheimer's disease, but tau, while it is very important in Alzheimer's disease, but independently also causes several neurodegenerative diseases. We call them collectively tauopathies. Okay, so for that reason, we decided to target mostly A beta. And you see, this is a co-stained sample, and there is no overlap. That means it's perfectly it's very, very selective to A beta, and it perfectly distinguish Alzheimer's disease from tauopathies. And of course, I am not showing data here. Probably in the next slide, I might show you. So it also distinguishes Alzheimer's from Parkinson's disease and few other neurodegenerative diseases. Now you might ask why that is so important. Well, if somebody has a dementia, more often it will be a mixed dementia, meaning so Alzheimer's can be coexisting with Parkinson or some other neurodegenerative diseases. So unless you find out, identify what is what, so no way of even thinking of a therapeutic development. So here we have a probe, so it can, very effectively distinguish Alzheimer's disease, detect Alzheimer's disease, but also distinguish Alzheimer disease from other neurodegenerative diseases. This is again, let me tell you, this is a very, very unique probe. Of course, probably I'll skip here. This data shows that the selectivity of the probe, for A beta. And of course, I would like to highlight, rarely we come across small molecules which outshine antibodies. Okay, I am sure you all agree. When we talk about some kind of biomolecular interaction, antibody-antigen interactions are always a gold standard, and especially I know we always like to go with the antibodies for any kind of studies. So here we have a small molecule which has both in terms of affinity and selectivity, it is all matches antibody credentials. So here is again data from the AD brain sample, and you see co-stained with our probe and also with another probe which is commercial available. It's called thioflavin. I would like to mention to you that we can actually force any protein to undergo aggregation. Okay, I hope you are all from life science background, you agree with me. And this thioflavin, it's a very cheap and commercially available, can bind to all that any kinds of protein aggregates. Okay, so it is not specific at all. And also looking at the image here, you can see that there is so much of background, and you see that is our probe, so clean. And that is the merge, the image, you can see the dots. That is the selectivity. It shows the selectivity because our probe binds to specific conformation of of this flux. Of course, as I mentioned, I will not again, probably I have to speed up now. So, for example, our probe can also detect this, a congophilic A beta deposits, which is called congophilic angiopathy, meaning where these A beta are, deposited around the blood vessels. And this data is actually from, AD patients as well as from the patient, brain samples, which the patient has a supranuclear palsy, which is one of the tauopathy, which clearly, kind of emphasize or, reiterate that this probe is very, very selective for Alzheimer's disease. Well, that is a ex vivo. Of course, they are all human AD samples, but it is done ex vivo. And for a real life applications, we need to show that indeed they work in vivo application. Of course, we cannot work with the human samples directly, that you are all aware. So instead, we can actually work with the mice models. Now we have a double transgenic mice model that has been established in our lab here in JNC, and it's a very, very unique model in the country. And of course, because there is a reasonable level of the matching with AD brain and human brain, and if you can prove that that this probe works in the human AD brain, probably we can at the AD mice brain, we can take it forward for human clinical applications. So that is the data I showed you. That is our probe which detect AD, it's a flux or Alzheimer's disease, ex vivo. And now, as I mentioned, so we have kind of set up this double transgenic AD mice. And these mice, luckily, our probe, I'm not showing the data, our probe crosses blood-brain barrier. So that was one good thing. And we have treated mice, wild type and AD mice, and you can see that the wild type has do not have much of the flux, but the one with AD phenotype, you can see that so there's a quite a bit of A beta A beta flux. And again this data also make it clear. See, that is our probe, so clean, it very, very selectively detect A beta. But when it goes to thioflavin, which I told you, which is it's not a selective probe, you see so much of background. In fact, it's now kind of confirmed that it's a very selective and in vivo probe. And of course, we are also now taking it forward, for making it a PET probe and also near infrared probe for clinical application through our own startup company. So that I will talk about it, and I'll mention later on if required. So hopefully we can convert this into a diagnostic probe for in vivo application for early diagnosis of Alzheimer's disease. Now coming to reactive oxygen species, I have mentioned reactive oxygen species. They play a very, very important role in Alzheimer's disease. Okay, so in fact, few years ago, when we proposed from our lab that reactive oxygen species plays a very important role, we had a tough time in convincing referees and editors because they say reactive oxygen species are also, implicated in cancer and many other diseases. But later on, several studies from, from our own group and other groups, now it's possible, people believe now that indeed reactive oxygen species plays a very, very important role in the form of oxidative stress and neuroinflammation in AD. And in fact, in one of the paper, some other group after our work also, they say that plays a unifying, mechanism in neurodegeneration and also become a kind of a drug target. So, if we can, tackle this reactive oxygen species or related oxidative stress and neuroinflammation, that can be a potential therapeutic for Alzheimer's disease as well. Okay, since I mentioned about reactive oxygen species, let me spend probably 1 or 2 minutes on that. So reactive oxygen, the moment I say reactive oxygen species, so please do remember they are important, for physiological, functions. They are very important. That's why I say they can have a life-saving effect, but they can also have a threatening effect. Meaning, you consider these are a tiny species. These tiny species, for instance, the oxygen undergo 4 electron reduction to form a water. And in the process, it goes through superoxide, hydrogen peroxide, and hydroxyl radicals. These are the highly reactive oxygen species. They are important in our body for physiological function. However, if their concentration levels goes behind a threshold, they become extremely toxic. Let me tell you how extremely toxic they can be. You might have seen a kind of a dreaded a very biggest probably dangerous villain in a movie or a drama or in your dream, or you can imagine. Okay, but these tiny species, there can be no bigger villain than these tiny species if they cross a threshold in our body. Okay, so among all these reactive oxygen species, hydrogen peroxide is one of the reasonably long-lived species, and this, in presence of myeloperoxidase, convert to hypochlorous acid. I hope many of you are aware. Okay, if not, probably I'll try to tell you briefly few things about it. Hypochlorous acid is very, very important. In fact, it is part of our immune system. Okay, to make it more simpler, hypochlorous acid is basically a bleach. The bleach which we use for cleaning, so that is hypochlorous acid. Of course, sodium or a calcium, salt of it. And I'm not sure how many of you are following in this COVID-19 situation. There was also a bleach controversy involving American President Donald Trump. Okay, so I'll not go into the details. If you know, some of you probably you can recall. So this hypochlorous acid is very, very important. Okay, so in fact, this is the one involved in it's important in our immune system, because this actually can involve in digesting or deactivating pathogens such as bacteria and viruses. So that's why it will have a life-saving effect. Okay, it is produced in the phagosomes, okay, or neutrophils, and then it digests bacteria and viruses. Okay, and it's so very important so I'm emphasizing this because it's very important. If somebody having a immunity against COVID-19, I think you must be assured that you have quite a bit enough of this hypochlorous acid. Unfortunately, that while it is it is it has to be produced endogenously, so you cannot drink bleach to cure COVID-19. Okay, that is very, very dangerous. Okay, so coming back to Alzheimer's disease, under Alzheimer's conditions, so there is over expression of myeloperoxidase that converts lots of hydrogen peroxide into hypochlorous acid. See, as I told you, as long as the concentration of these reactive oxygen species within a limit, it's good. I mean, they have a life-saving effect. And when they're produced in excess, excess, so they will have life-threatening effects. Okay, so they create responsible for oxidative stress, inflammation, and including DNA damage, lipid damage, and lipid peroxidation and protein oxidation. Altogether, they contribute severely to progression of Alzheimer's disease. So in fact, among these reactive oxygen species, as I mentioned, hydrogen peroxide and hypochlorous acid are slightly long-living. So we, in our lab, we developed a very interesting probe. For example, this is called QCYBA, a near-infrared probe for detecting this hydrogen peroxide. So this reacts with, very selectively with hydrogen peroxide to generate this probe, this molecule. This is, on binding to DNA, it gives a near infrared probe. In fact, I'm not going into the details. If you are interested, you can look into these papers. Okay, so literally, this is again a very unique probe. Using this probe, you can monitor hydrogen peroxide in a cell, especially whether it's its levels, the homeostasis of hydrogen peroxide, its production, its degradation, everything can be monitored, beautifully. And of course, we are also trying to use this probe for assessing hydrogen peroxide levels in case of Alzheimer's disease. And very recently, so we developed a very simple probe, it's called CM1 probe, which is a coumarin. He says a little bit of chemistry. Don't worry, I will not talk too much of chemistry here. So coumarin probe, which is a with a coumarin is attached to a morpholin. Okay, so this is an amide probe, and which we converted into a thioamide, and that become non-fluorescent. Okay, this is fluorescent and that is non-fluorescent. And this thioamide probe, in presence of hypochlorous acid, either in vitro, in cellular, or in vivo conditions, selectively transform to a amide probe, and which is fluorescent. So it's like a off-on signal. So this is a off state, and in presence of this hypochlorous acid, that will turn on to a on state. So that's all you have to remember. And of course, this reaction or a transformation is very, very clean, as you can see here. And it's also very selective. Well, in our, in the cell, there are so many reactive oxygen species, many of them. And then we also tested them, and it is very selective to hypochlorous acid, and also reasonable good, good quantum yield. And encouraged by this, we also tested their, cytotoxicity. This probe is not toxic to cells, and it also exhibited good cellular uptake. And then we went ahead and we tested in the cells, see, whether it can detect this hypochlorous acid. So these are the cells, control cells, not much, signal. And we treated the cells with sodium hypochlorite, externally. And then we treated with our probe. You can see that the hypochlorous acid is generated in, can be detected easily. So next, what we did was, this is interesting experiment. We treated the cells with A beta and copper complex, amyloid beta and copper complex. If you remember, the redox metal can form an inclusion, can bind to this A beta and become, generate, it become redox active and generate reactive oxygen species. So here, the reactive oxygen species are generated in situ, and of course, HOCl is also part of it, and that can be detected very nicely. So this was very encouraging results. So based on this, so we tested whether this probe can actually cross the blood-brain barrier. Indeed, it crosses the blood-brain barrier, and the mice were treated with this with the probe, both wild type and AD mice, and we can beautifully detect this hypochlorous acid in the brain. What is more important or interesting is that the red signal you see, that is all for that is from the A beta, and the green signal you see, that is from hypochlorous acid. So if you carefully look or quantify, you will be able to see that hypochlorous acid that we are detecting is actually proximally produced by these A beta aggregates and proximally localized with the A beta. And then of course the quantification data also shows both in the cortex and hippocampus, and so that the hypochlorous acid is produced and proximally localized with A beta. So meaning hypochlorous acid plus A beta together forms a perfect biomarker for Alzheimer detection. So that's what. So here I had two things we achieved. Okay, for you. So what I showed you, this, you see this image. So it's not just image. In fact, you are visualizing multiple toxicities of in the AD brain. Although I mentioned only about A beta, I mentioned about ROS, but you must also know that that triggers oxidative stress, biomolecular damage, mitochondrial dysfunction, and also neuroinflammation and many other aspects. So in a way, so I also, this image also shows that there is a multifaceted toxicity in Alzheimer's disease. In addition to that, we also validated a new biomarker combination, okay, which now qualifies to be added to the list I mentioned, NIA, National Institute on Aging, Alzheimer Association Research Framework 2018. So the definite biomarker list I mentioned earlier. So this biomarker combination or a pair qualifies to be a definite biomarker, can be used for Alzheimer disease, detection. Of course, we have developed biomarkers and methods and protocols for detection of variety of biomarkers, biomarkers which have direct and indirect implications. Ultimately, our idea is to multiplex them, multiplex them to kind of generate that kind of a fingerprint, such that we can easily distinguish from AD patients from healthy individuals or individuals with some other symptoms. So that is the idea. And of course, so just since it's it's not a, please don't consider this as an advertisement. So since yours is a life science department, and also in line with Atmanirbhar Bharat and Make in India, so we have our own startup company that is called Veneer Biotechnology Private Limited. So wherein we give a solution, bioimaging and diagnostic solution. In fact, we have kind of a range of molecular probes, both from our own lab as well as some of the commonly used molecular probes, which you all use, which are easily accessible compared to, see, most of the time life science in our labs. So we procure these probes from abroad. It takes time consuming. Of course, they are expensive, and so on and so forth. And here we have range of molecular probes, almost all molecular probes which are used in biology, in a life science or biochemistry or chemical biology labs. And if you are interested, if you want some probes, please do write to us. Sometime we should be able to support you in any way. Even if you are looking for some solutions, that also possible. We can provide you a solution, in both in terms of imaging and diagnostics. Okay, so far I talked about.
Govind Raju started screen sharing. Start aita.
Huh?
I would, I'm so sorry, sir. I would like to request the audience member to mute their
Hello.
mics. Miss Lavanya Reddy, can you please mute your mic? Yeah, thank you. Sorry, sir. I think a few
Okay.
of them are okay. You can continue, Dr. Govind
Okay.
Raju.
How much time I have?
So you can speak, sir.
Okay. Okay. So thank you. So far I introduced you Alzheimer disease, as a multifactorial disease. I also introduced you. Present to you that how we are working on working towards developing a diagnostic protocols or kits for Alzheimer disease. Okay. So now I will quickly take you through our through various efforts that has been going on in our lab in terms of developing therapeutic candidates, drug candidates for Alzheimer disease. As I mentioned to you, so we started working on Alzheimer disease some years ago, especially 10-12 years ago. And when we started, so we also kind of new to the field, and we were also kind of thinking, we thought about individual pathway. Not that time we were also not clear about the complexity of Alzheimer disease. So we also decided to target individual toxicity pathways or routes. The first thing comes to our mind is, why not we kind of work on some of these natural processes which can clear these toxic aggregates from the brain. And of course, one is that proteasomal degradation, which we discussed. The other one is autophagy. Okay, autophagy means self-digestion. It literally means self-digestion, meaning this process. For example, if you are starving, so these autophagy get activated so that it digests some of the organelles, so that supply energy, energy for the cell. And also, if there are dysfunctional organelles or proteins in a cell, so the autophagy is supposed to actually kind of remove them, clear them from the brain. For some reason, of course, with that the process got this discovery and understanding of mechanism of autophagy got Nobel Prize in 2016. Okay, So by then, we were also already working, we have published our papers, we actually started, around 2010, a project on this. So, and this got Nobel Prize. And in fact, we developed a library of compounds. Again, I'll not go into the details. We developed a library of compounds and we screen them. And in fact, we identify a compound which can enhance autophagy. Okay. So that encouraged by that, we went ahead and we developed a few more, peptide kind of molecules, hybrid, modified versions of the peptides, and which indeed shown to have very good modulating effect on these A beta aggregates. For example, these are the, we have studied in the yeast cell. See, these are the yeast cell. You see that, they going happily. And the moment you express this A beta, moment you express this A beta in the yeast cell, so their growth has been retarded. And these cells which are, affected by A beta are treated with the one of the, the lead peptide, and which actually rescue, okay? This rescue these neuronal, rescue these yeast cells from A beta toxicity. I think seeing is believing. You can see that here is the yeast cells, and you see the green dots, they are the A beta aggregates in the yeast cell, is a model system, okay? And when we treat with our peptide, okay, it goes and binds to it and then dissolve these A beta aggregates into a monomers, and these monomers will be engulfed by what is known as autophagosome. Sorry, I cannot go into the details. So, okay. So, and these will fuse to lysosome, and that will be again, degraded. Okay. That's how, it works. Well, this was very, very interesting and encouraging for us. But at the same time, at this stage, we realize, so targeting individual pathways has, no role. We will not going to help, dealing with tackle Alzheimer's disease. More than that, autophagy is a bit complicated process because it involved in both, healthy state, disease state, and also in many, other conditions. So modulating autophagy may be difficult. So we were at that stage, we started looking for multifunctional inhibitor, meaning a small compound which can target more than 2 or 3 disease roots or toxicities. So that's what the multifunctional inhibitor or a molecule or a modulator. Same molecule should be able to clear A beta aggregates or prevent aggregation, and it can also for example, it can act as an antioxidant. It can ameliorate the cells from neuroinflammation, and probably some other things as protect mitochondria. It can protect biomolecules, all that. So such molecules are multifunctional inhibitors. So we at that point we decided, designed a hybrid multifunctional inhibitor to start with. Meaning, see, this is the compound. This is called cleoquinol. Okay, so very simple compound. So it also has some applications in what is called this veterinary science and things like that. Again, I will not go into the details, but nevertheless, this cleoquinol and its generation 1 and generation 2 compounds were the first to enter clinical trials for Alzheimer's disease. Okay, so there were good outcome. However, it is failed in one of in the advanced clinical trial because of the toxicity. In addition, here is a compound called EGCG, epigallocatechin gallate. Okay, so this is EGCG is a polyphenolic compound which is there in our green tea. Okay, I'm sure many of you are all of us we take tea. Okay, green tea, and then nowadays green tea has become fashionable, so it is there in green tea, and everybody recommends to drink green tea. Okay, and this is one of the constituents of green tea, and this has been tested for AD, and it's also under clinical trial. But let me tell you, while this cleoquinol activity was very high, but it was toxic. But the epigallocatechin is not toxic, but the activity is very, very slow. Okay, meaning because it's more of a nutraceutical. See, you can, if you drink a green tea for a lifetime or a 10 years, maybe you may see some beneficial effects. But when we talk about a drug or a medicine, so it has to be it has to work in a daytime, in a day or 2 or a week time or a month time, or at least a year or a 2. Okay, you cannot wait for a lifetime. So in my opinion, this cannot be a drug candidate or a medicine. Okay, so it is more of a nutraceutical. So what to decrease the toxicity of this compound and to increase the the activity of this epigidocatechin, we decided to integrate the structural and functional components of these molecules, and we derived these hybrid multifunctional compounds. And in fact, one of the compound was found to be relatively non-toxic in the cells. And we went ahead and showed that indeed this compound is a multifunctional compound. It can actually ameliorate many of the toxicity which I talked about earlier under Alzheimer's conditions. Of course, we also did some other molecules also in this direction. But at that point of time, we come across this peptide. I hope you all can recognize the peptide. This is a short tripeptide. Okay, there are 3 amino acids: histidine, glycine, histidine, and then lysine, a tripeptide. Okay, so this is there in all of us. It is there. You have it, I have it, everybody has it. Okay. In fact, when I give talk to chemists, I say that it's a natural product from humans. This is a wonder peptide or a tripeptide, because even sometimes I don't mind calling it a Sanjeevani, because it does so many functions in our body. It's very, very important. So it does all that functions by complexing with the copper in our body. So it has kind of antioxidant property, anti-inflammatory activity, and also has a kind of a gene regulatory activity. It helps in wound healing. I can go on and on and on. List so many. Okay, that's why I told you it's a wonder tripeptide. All of us have. There are 2 points I would like to mention. See, adults. So most of you, the students who are attending, you will have high quantity of this. Usually you'll have 200 nanogram per ml body fluid. But as we age, the concentration of this goes down even to below 80 nanogram. So that means that is directly linked to some kind of aging and or disease conditions. In addition to that, this can complex to this copper, as you see here, the complexation, the KD. If you consider a KD values of this complexation, this value is such that it can easily sequestrate the copper from A beta. You remember I mentioned copper can form an inclusion complex with the amyloid flux. So this can easily take out the copper without affecting enzyme, because we also have many enzymes based on the copper. Okay, so that should not be affected. So this KD value is perfectly matched. Is such that it can take off this copper. Okay, copper such that that can stop the production of reactive oxygen species without affecting enzymes. Okay, considering these parameters, so in fact we designed. So this was the peptide I present to you. In that ATAPAG we have used. Okay, we made this hybrid peptide, and in fact this turns out to be a multifunctional inhibitor in case of Alzheimer's disease. That was wonderful. So it can do so many functions. I think you can read them all. I already mentioned earlier. That was very nice. So it was excellent, but there were 2 points to be considered here. So it works as a multifunctional inhibitor, but it works at above 100 micromolar. That was too high. And in addition to that, it also functions as a multifunctional inhibitor only in presence of copper. But if you remember, I also mentioned there is also iron. The ROS is also produced in presence of iron. The ROS can also be produced in the independent of these metals as well. So we need to tackle them as well. So those were some of the disadvantages. So being we also have having the advantage of chemistry background is also useful, especially organic chemistry. So we did some kind of surgery here, and then we come up with these lead molecules. And in fact probably I'll skip this synthesis. Yeah, so these molecules were now found to be the GHK binds only to copper. Okay, see, for example, our molecule and GHK, they all bind to copper, okay, including parent. But when it comes to iron, the GHK doesn't complex. So only our molecule. Suppose this is also characterized by many other techniques. And then we did an assay called Trolax assay to determine this oxidative, antioxidant property. In fact, it does excellently well, all the compounds, and among them, this particular compound 4 was found to be the best antioxidant. And it also, we tested this anti-inflammatory property in glial cell. I hope you all know that earlier everybody thought only neuronal cells are important. But now, and all other cells, glial cells are only supporting cells, but that no longer true. So glial cells also have a very, very important role in Alzheimer's disease, or in other words, or even in healthy brain. They have a lot of functions. So inflammation is very, very important. And in fact, one of these, the glials, the role of glial cell is one of the hot topic in Alzheimer's disease. So in fact, our studies also showed that our compounds, behave, function as an anti-inflammatory compound. To test this both anti-inflammatory and anti-activity, in a realistic scenario, we followed this NRF2 pathway. NRF2 is a transcription factor. So whenever the cell is under oxidative stress, so this is NRF2 is mostly in the cytoplasm. And when you, for example, you subject a cell, when cell is subjected to oxidative stress, this translocate, NRF2 translocate to nucleus, and then it binds to a DNA, and then initiate transcription of anti-oxidative and anti-inflammatory genes, so that this oxidative stress can be taken care of. Now we decided to test what would happen in presence of our compound. See, these are the standards, the healthy cells. Okay, you see the green signal, whatever you are seeing, that is actually from the NRF2. Okay, you can see that NRF2 in the nucleus and also in the cytoplasm. And you subject these cells now to a ROS, okay, reactive oxygen species, and the entire the NRF2 is translocated to nucleus. But if you do the same study, okay, if you subject these cells to reactive oxygen species in presence of our compound, for example, compound 4, you can see that NRF2 is still there in the cytoplasm, meaning our compounds are actually taking care of this reactive oxygen species without the requirement of NRF2 translocating from cytoplasm to nucleus. So that clearly indicated that indeed this compound, our compound, can be multifunctional inhibitor, and may have useful, it can be developed into a therapeutic candidate. What I showed you is for example, the cells are under stress, okay, under multifunction, multifaceted toxicity under AD conditions, and in presence of our compound, so it actually rescue these cells. Of course, so we went ahead, and then we so in such kind of diseases, one kind of effort will not be useful enough. So we need to try many different routes, okay, and strategies. So here is another strategy. So we have developed very recently, we developed a polymer, okay. It's literally a polymer. We call it polyampolite. We call it polyampolite because it has both positive and negative charge. It can behave as a synthetic protein. It's like a protein. It can behave like a protein. In fact, that's what it happened. See, for example, this is a lysosome. You all know that if you subject lysosome, to eating, okay, temperature, okay, so that can undergo aggregation. But if you do the same thing, in presence of this polyampolite, see, that is been protected. In fact, it behaves like a synthetic chaperone. So this polymer can protect the aggregation of aggregation or a collapse of 3D structure of a protein. So in addition to, so that's what of course is also prevent A beta aggregation. And more importantly, so lithium has been used in many as a neuroprotecting agent. Well, many of its effects are not very clear, but nevertheless, lithium has been used in many of these neurological conditions, to overcome depression and things like that. And we but however, they use kind of several milligrams of this lithium salt, okay, so that will have a lot of side effects, definitely. So we are this particular polyampolite not only function as a protecting the structure of protein, but it also can kind of encapsulate lithium, and then it can deliver lithium into the cell. And we all know that as it goes to, for example, it goes through endocytosis. So there the pH changes from neutral to acidic pH. Under acidic pH it can release. So here this will become a it's like a dual function. It's not only modulating protein aggregation, it can also deliver lithium therapy. So it's a very recent work. Hope we believe that people will take cue from this and develop some therapeutic candidates. As I mentioned, so one strategy is not good enough to develop that. So of course we work, as I told you, I showed you small molecules, I showed you peptides, okay, I also showed you polymer, and we also worked on natural, looked into natural products. Okay, here is in that natural product, here is a compound called berberine. Okay, berberine, it is like a curcumin. In fact, it looks like a curcumin, by color, okay, and material touch, it looks like a curcumin. I am sure you all know that curcumin has been tested for almost every disease, okay, as a inhibitor, as a drug. So, and so is the berberine. Berberine has also been tested for many diseases. Unfortunately, so also this is berberine is toxic to cells. Okay, this is something people not talk about. It's toxic to cells because of its solubility. It's less soluble. In addition to that, it also brings about what is known as mitochondrial fragmentation. So that is the toxicity mechanism. So to overcome that, we modified the structure of berberine. That's what called berberine D. That's also mentioned in the when introducing. Okay, see that is the natural berberine natural product is toxic, but the BERD, the modified compound, is relatively, or in fact, is almost like non-toxic. And we went ahead and then showed that in fact it protects. So mitochondria, I'm sorry that I'm not going to the detail what is this protection is all about, but this data indeed shows that it protect. There is, for example, these A beta oligomers can actually pinch holes into this membrane, and then they change their the potential, and also reactive oxygen species generation and so on and so forth. So it protects not only both cell membrane and also mitochondria from that, and also in doing so it rescues cells. So this was confirmed by by following the apoptosis markers. I am sure when the cell goes to apoptosis or a death, so this there will be a increase in the levels of caspase and cytochrome C, and that's what we have followed. So in the presence of A beta, so lots of this caspase and cytochrome C are produced. And when we treat these cells in presence of our, with our compound, a candidate, drug candidate, you can see that the levels have been, it's not completely diminished. That is also not expected. So the levels have been brought down to a healthy conditions. In fact, it's confirmed that indeed it is a multifactorial compound. However, well, this is excellent. We have an excellent result from this, but there is one problem in this particular compound. To take it further for animal in vivo studies, meaning it doesn't cross blood-brain barrier. Okay. For that, of course, we have expertise. We are trying to use nano or bio-nanotechnology techniques to study further. So that is ongoing work. To conclude, the last portion, probably last 2 minutes, maybe a couple of minutes, I will try to finish. So of course, we also kind of now identified a drug candidate purely by design. Okay, this is not by natural product, it is purely by design, a synthetic compound. So that is the one which is mentioned here. Of course, we have made a focus library of compounds. We screened them and we identified this compound TGR63, which is actually is there in the bioarchives. If you are interested, you can look into that. Let me tell you that this is a 9 years of work. Okay, it's not just 1 year or 2 years. So this is a 9 years of laborious work. So finally, we are happy that we could conclude this work, including in vivo studies. So these are all some computational studies also showed that, in fact, this compound can inhibit, bind to A beta aggregates and it dissolve them into monomers, and it also can prevent the formation of aggregates, by binding to a monomer itself. And this is the data shows that, for example, these are the A beta aggregates. When we treat with our compound, it prevents the aggregation, and of course, if we also treat with aggregates, it can dissolve them, and more importantly, it protects the cell. Okay, so this data tells you that indeed this compound protects cells from A beta toxicity. Of course, these are some of the mechanism how it does. So here is the data. So seeing is believing. So again, we know it's quite a bit of work. I'm condensed here. So we went ahead. So after we got we got a good this gave us the confidence to go for in vivo studies. As I mentioned, we have developed a mice model. I hope you are all from life science background. So maintaining AD mice is one of the toughest. AD mice is one of the toughest mice to kind of maintain. Very, very difficult mice. So we are one of the unique lab in India to have this established mice model. And you see, this is the AD mice. Okay, so not much of AD flux. And this is the this is the wild type, and this is the AD mice. And you can see that quite a bit of huge chunks of A beta, okay, in the AD mice. And now we treat our mice, these mice with our probe, so our drug candidate. You see that so drastic reduction, quite a drastic reduction. Okay, that is you can clearly see that. So that's how it is reduced. It reduces the amyloid burden. So our treatment of AD mice with our compound, okay, drug candidate reduces this amyloid burden drastically. That resulted in very, very interesting. It also not just reducing this A beta burden, that also resulted in very good improvement in terms of cognitive behavior. Of course, these are the A beta flux in the vehicle, okay, untreated mice and treated mice, okay, in the cortex and hippocampus region. And of course, we established how not only just seeing kind of a histopathological data. So we also showed that indeed the reduction in A beta and this amyloid flux result in improvement in cognitive deficits. We have done three, not just one, three different cognitive tests. One is open field test, another one is novel object identification test, the third one, and also most important one, Morris maze water maze test. Okay, all of them have established. So data is here, so I will not go into the data. I will not going to explain to you the data, but I will show you one video and that will be will clear to you to how effective our drug candidate is. And of course, if you are interested, as I told you, please do go into you can look into this bioRxiv paper, where we have not only the data, but we also have 3 different videos which for each one of these behavioral tests, which tells you how good this drug candidate is. But today for you, I will just show you one video so which will have clearly explain to you or show you the efficacy of our drug candidate. So let me go out and for a minute so and then upload this video. Okay, can you see the window?
Yes, we can see it.
Okay.
Morris water maze, right?
Yes, wonderful. Okay, let me okay, so what you see is the Morris water maze test. So what we do is you'll have a water tub, tank like this, and then you'll have a platform. You can see that you can divide this water tank into a 4 quadrant, and in one of the quadrant we'll keep a platform. Okay, and then we leave the mice with and without AD symptoms or a phenotype, and then you see how quickly they can identify. That is called a learning, learning. And subsequently, what we do, we remove this platform and see these mice, how these mice can actually remember the position of this platform. So that will be a memory. So together, it will tell us their cognitive, levels. Okay, so that's what I'm going to show you. Here we are. You see this, there is a wild type mice here. Can you see my cursor?
Yes, it is visible.
Wonderful. Okay, so this is wild type mice. Okay, and then the wild type mice, they are healthy mice, and here is a healthy mice, and here is a healthy mice treated with our compound. So just to see that is there any toxicity, okay, or a side effect on the healthy mice. And here on the right side you have a AD mice which is not treated, and then the top right side bottom corner. So we have AD mice which is treated with our drug candidate. Okay, and you see the circle, that is the platform, and see how quickly they identify, they find the platform. So here is a healthy mice right away. Wow, see, that found the platform. And the wild type as well, wild type treated with the drug candidate. And these both AD mice still finding the platform. You see, the AD mice, which is treated with our compound, already found the, finally found the platform. But this other one, untreated one, is still roaming around, all the way. Okay, so that clearly tells you the difference between healthy mice and AD mice, and also the effect of our compound. So next, of course, as I told you, I will not go into the data. So here you have removed this platform, okay, and see whether these mice remember, okay, whether they have the memory, okay, they memory, whether they have a good memory to remember the position of platform, okay. See, these healthy mice, they are always roaming around here because they know the platform was here, okay. And so is the treated one, Alzheimer mice treated one, but this has no clue. Okay, so this is a very scientific, way. It's not just about a video. So these are all the data. So again, I will not go into the details. So it is, there is a quantification, okay, on this data. I hope it's clear to you that indeed our drug candidate is actually, rescuing the mice from AD conditions, both in terms of A beta burden as well as a cognitive thing. Okay, where the slide? I think I missed the.
You have shared only this screen, sir. I think Professor Govindarajan, you'll have to go back to sharing.
Yeah, I just went back. I am not. Oh, here. Yeah, okay. Stop sharing.
Okay.
Yeah. Okay, so I'm almost there. So that was the story of our 9 years of one, kind of chasing one molecule, or one project to identify a drug candidate. So at least in the mice model, we have identified, with the confidence, we can say that we have identified a drug candidate. In fact, what I didn't mention, that this is like almost, the test. So we have tested this in the early stage, mild cognition, and also advanced stages of AD conditions. So interestingly, our compound works in all the stages. So to conclude, hope I could convince you that Alzheimer's is a kind of a devastating disease, and because of its multifactorial nature or a multiple toxicities, and I also that means it involves multiple biomarkers and targets. And I also showed you our own results. So for where not only just detecting Alzheimer's disease, but also differentiating it from other neurodegenerative diseases, and also kind of discovery or identifying a novel biomarker which can be used for clinical diagnosis of Alzheimer's disease. Overall, the multi-pronged approach, which is uniquely, it is very unique that we are kind of following, is the potential to deliver diagnostic and therapeutics for Alzheimer's disease. And of course, finally, I showed you, of course, the drug candidate which we have identified now, which has at least reached a mice model, very effectively. And of course, way forward is, as you remember, we also work on both diagnostic and therapeutics, and we are also now looking towards diagnostic therapy, which is something upcoming, not necessarily already is there, so which is called theranostics. So to have a kind of a candidate which can be used for both diagnostic and therapeutics. So before I conclude, I would like to thank my students So here I would like to, particularly for the student community, I would like to mention that, ours is a, as a, you all, your teachers, will tell you that no more boundaries. Okay. So that is truly what's good in our lab. So although our lab has started as a, kind of a bioorganic chemistry lab, but now, we have, kind of conquered all these boundaries. The student you see here, or some you may not, in the picture, okay, at least in the name. So they are all from all kinds of backgrounds. We have student from organic chemistry background. We have student from biochemistry background. We have student from microbiology. We have student from biotechnology, and we have student from, in fact, MUSC, okay, veterinary science. So we have, in principle, we have, kind of a group members, from all kinds of background. So I would like to thank all my students, for their hard work, and especially the work I presented is mainly done, started by Rajshekhar, who is working on human biome project, in Switzerland now. And then the Madhu, he's a doctor, veterinary doctor, MUSC doctor from IVRI, well-known veterinary institute. And Saurav Samanta, who is the last mice work I told you, that is the work established by Saurav Samanta. He has just, is about to become a doctor mostly. And of course, others, we have so many other ongoing projects. So many of them are involved in some of those different projects. And also I would like to thank some of our collaborators. Okay. So thank you all for your kind attention. That is our lab, one of the main lab in JNC. If you happen to visit, so I'll be happy to host you anytime. Okay, after COVID-19. Okay, and then we have both, kinds of biology lab, cell culture lab, for life science, doing all biological studies. And of course, we also have an animal house where we maintain animals for not just for Alzheimer's, for also others. Of course, Alzheimer's is one of the major mice model. So thank you very much, and I will be happy to answer if you have any queries or comments, questions. Thank you.
Yes, thank you so much, Professor Govindaraju. It was a really insightful talk on the Alzheimer's disease as well as its probable amelioration using your natural as well as the synthetic, drug candidates, as you call them.
Thank you.
Okay, and I'm sure that our students as well as the other members of the audience, everybody who's present here has benefited immensely. And I would like to welcome some questions in the chat box. For now, let's see, we have one question I see. I'll just ask you that.
Sure.
So it was an interesting, Violina is asking, she's a student of ours. Sir, can you tell how to distinguish Alzheimer's disease from other degenerative diseases? And what are the unique symptoms to detect the AD in an early stage?
Okay, so that's a very interesting question. So probably, I was quick, when I was talking about that, probably I was quick. I didn't delineate too much. So you have kind of two parts to your question. So first one is how to distinguish from different neurodegenerative diseases. Well, as I told you, currently, in a current diagnosis is mainly based on the behavioral symptoms or a cognitive deficit. Meaning, for example, when it comes to Alzheimer's disease, let us say in one of our own family member start forgetting things. Okay. So, forgetting means, we all forget, this is what happens when I say, that's why, in fact, I had a very nice slide I just avoided because not to scare you. Okay. So when I say forgetting things means it's very common. Okay. We all forget. So, but you forget something and then after a while you remember. So that is the normal thing. But some of our members, family members, probably when they cross 50, 55, okay, so they forget, they will have, forget, and then they will never be able to recognize back, okay, and then they will have a problem in doing day-to-day activities, okay. These are, I'm not going to full details. See, these are all some of the activities, and then you will think, start thinking there is some strange thing about this, particular member, okay, and then you tell them, they will not agree with that, okay. So they may not be even aware that, something like this is happening. Okay. Some may, but some, many, they may not agree. So these are all the symptoms. And when you go to, many a times in Indian context, we don't even bother, by the way. Okay, that's what I said. The mental health is largely, neglected in India. Thanks to, I mean, at least now there is a, awareness. There is, awareness is coming in our public. But if you go back, 5 years or 10 years, I think absolutely there was no awareness in terms of mental, health. So when somebody just they may ignore, but sometimes when somebody is aware, so they will probably consult a doctor. Okay, a doctor, and then definitely when they go to them, they will do set up simple this kind of testing their memory and things like that, and also there are internationally recognized some kind of objects, these, that, and all, the clinical doctor, so we will see and then they will identify. But this is the way currently being diagnosed. For example, if you look at the Parkinson disease, so there will be tremors. So you find that you don't find tremors in case of Alzheimer's disease. Okay, you can distinguish partially, but this is not the right way of doing it. That's what I mentioned. If at all, if you want to go for a proper diagnostic, I think we need to detect those biomarker which I mentioned. Then only we can say that the proper diagnostic. So far, at least elsewhere it is started happening. So in India, still we don't have it, but hopefully, if not, immediately, at least 5 years or 10 years from down the line, we will have this definite detection. Okay, so definite diagnostic. And then coming to distinguishing also, I said use the word dementia and mixed dementia. Okay, so more often when somebody has this dementia, when I say dementia, again, is a cognitive decline. Okay. And this dementia can be anything, not just Alzheimer. Okay, it's just that Alzheimer constitute 70 to 80% of all dementia, but other 15 to 20% also can be, for example, Parkinson. That can be from ALS, and which Stephen Atkins had, and it can be something Huntington, and there are a few other tauopathies, all that. So I don't know whether you have heard of a disease called Parkinson plus. Okay. So it's something called Parkinson plus, which is equally very devastating. So where both Parkinson and Alzheimer coexist. Unfortunately, it's not easy to distinguish. But for example, the method I kind of showed you, these are all some of the method which will be very handy. Okay, in detecting them. Okay, there is Alzheimer's disease, there is also a Parkinson's disease. If you don't do that, then the molecular mechanisms are slightly different in case of Alzheimer and Parkinson's disease. So how do you, if you don't distinguish, how do you treat them? You may be treating for Alzheimer, then Parkinson will remain. Okay, and that aggravate the disease. Okay, that's what I meant. Yeah.
So we have one more question from our student Sagar. He's asking, sir, as AD causes damage and death of brain cells, what is the most common cause of death of an AD patient?
Okay, so it's again a good question. So when it comes to, for example, in the last stages, when somebody dies by Alzheimer's disease, not necessarily it's like so he'll be dying just purely by Alzheimer's disease. Okay, so that once in the advanced stages, that can be leads to many symptoms. So I'm not getting the term. Like for example, pneumonia. Okay, things like that. Okay, so it also can be leads to symptoms different from Alzheimer's disease. So yeah.
So we have one more question from our.
But the neurodegeneration is the major this one. So I also, because you are reasonably advanced life science students, so that's why I didn't show. So there is a drastic reduction in the brain size. That is because of the neurodegeneration. Okay, the neurons die. As such, also, even without Alzheimer, also the neurons die. As you know that as we age, even in the age, the brain size decreases. But in case of Alzheimer, that will be a very drastic. And for the same reason, if you remember in my first slide, in the functional amyloids I mentioned, we are using silk for biomaterial application. I indicated neurogeneration. So what we are trying to do is, I didn't mention, see, for example, if you identify a patient, he has Alzheimer's disease. So when we discover a drug, that drug is for the cells which are still alive under disease condition. By then, but the patient already has lost several neurons. So we are also working on possibility of stem cell implantation. So using this Silk scaffold, we have showed that stem cells can be, kind of a transdifferentiated into a neuronal cell. We believe that if we can implant the stem cells using this our Silk scaffold, so that can actually differentiate into a neuronal cell, and it can replace the cells which we have already lost, in addition to a drug discovery. Yeah.
Okay, so we have one more question from Dr. Vinita Balasubramania. She's asking, GHK as a peptide is associated with copper. Isn't this required for its stability? The levels of GHK is lower in older people because copper levels decrease with age. So she wants.
Well, I think there is a confusion. No, copper levels will not decrease with age. The levels of the GHK decreases. See, GHK is a good one. Okay, it's important for our body. So it has several functions in our body. Okay, that's why I use the word Sanjeevani. Okay, it does so many functions. Higher the levels of GHK, better is our health condition. As we age, so that condition it decreases. That means, so it there are many implications. I will not be able to go into the details here. What is happening? One more point I didn't mention. It is complexing to copper, means it complexes to copper and keeps this copper in a redox dormant state. Okay, meaning, see, for example, if you have a reducing agent, if you have a free copper, it goes to copper 1 to copper 2. It's a cycle. Okay, in the cycle, oxidative reductive reaction, and then it produces lots of reactive oxygen species. But this, on binding to GHK, that redox process is stopped. It's called redox dormant state. That means it stops the excessive production of reactive oxygen species.
So if I can ask a connected question, and copper does decrease with age. I have to tell you that copper does decrease with age, because even ceruloplasmin levels decrease with age. And,
okay, so
ceruloplasmin,
yeah, please complete.
Ceruloplasmin is an inhibitor of myeloperoxidase.
Mm-hmm. Yeah, so here I would like to differ from you. So not completely. Yes, it can decrease, but when it comes to Alzheimer, so there is an accumulation of copper, iron, aluminum, zinc. Okay, aluminum is a little bit controversial, but copper, iron, zinc, they are all accumulated in the brain. In the brain. I'm talking about in the brain. Yeah.
Yes.
Yeah.
So it is mainly a problem of clearance of different materials from the brain, right?
Yeah, in some sense, yes.
So it is more to do with the lymphatic system, right?
Yes. Yeah, in fact, it is. That's where it is. A lymphatic system is the most affected one, and
Yes.
that's where it starts. And the thing is, for example, if you look into Parkinson and all, you start with some region, and then still they remain. They don't spread so much. For example, substantia nigra and so on and so forth. But in case of Alzheimer, it starts from hippocampus, and that is part of the limbic system, and then it spreads to cortex, and it's become aggressive in the cortex as well.
Yes, but I think Parkinson's is quite distinct from Alzheimer's. It is.
It's a
neurodegenerative disease.
Which one?
Parkinson's is more motor neurons.
Yeah, motor neuron, but it's still it affects the neurons.
Yes,
and there is a protein called alpha synuclein, just like amyloid beta. So the alpha synuclein aggregation, it's in the region of substantia nigra. Okay, is responsible for this, and then that also leads to neurodegeneration.
Correct.
Yeah, but it's a very clear difference is there. Yeah.
Yes.
But the thing is, they can coexist, because we don't know. That's where I mentioned the mixed dementia. We can have the symptoms of both, but sometimes maybe tremors and all. You may say that Parkinson, but that person may also have Alzheimer or some other disease. So unless we have way to detect, so it will not be clear.
Okay. Thank you, sir.
Thank you.
I enjoyed your talk a lot.
Thank you,
Dr. Vinita Balasubramania, ma'am. Now I have one last question from our student here in the chat box. His name is Niranjan. He asks, does Alzheimer's disease occur in young adults? I think they're all scared to know if they can suffer from Alzheimer's.
Yeah. Okay, is again a very good question. There are probably, I'll try to keep it simple and probably in 3 points. Alzheimer, so whether it's, the question now comes whether Alzheimer is a genetic or a non-genetic disease. Okay. So Alzheimer is largely non-genetic disease, is a kind of a lifestyle disease, but there are genetic implications. That is almost 70 to 80%, I know, maybe little more than 80%. And there are 12 to 50% Alzheimer conditions which are directly linked to, genetics. That is, especially there are something called Swedish mutation, and there are families in Sweden, okay, and they get very early age. But all others, you and me, for example, in Niranjan and all others probably may not get it at the early stage, so you don't have to worry about it. But having said that, earlier it was definitely considered, it is a age related disease, definitely, there is no doubt about it. And for example, if you go back 10-20 years ago, usually the Alzheimer patients used to be kind of identified above 60-65, okay, that was the limit. But now that age has come down. Even I know current, just last year somebody died, he was 55 years old. In fact, the patient that Alzheimer who identified the disease, she also died at say 57 years age. Okay, this age is coming down. Probably it's also because of lifestyle, and there are also genetic. Although I said this largely non-genetic, but still we are yet to decide. We are also working on looking into various genetic aspects. But nevertheless, see, our physical and mental exercise is the levels are going down. Okay, see, now the question also is related to India. So earlier people were used to be very hardworking, and we didn't have all this kind of fancy stuff. Okay, so we have to do everything manually, and we are all healthy, probably not affected at the early, at least 70-80 years, if at all, if somebody leaves, life was not affected. And now, because of this all fancy thing and also biomedical research, our life expectancy has increased. In other words, because of this computer and this IT, all these things, our work style has changed, and the food, all this processed food, and it never there. See, we don't have a good mental exercise, we don't have a physical exercise. Okay, so that's where it's the number of cases are going up in India as well. So if you want to avoid Alzheimer disease, my advice to you and also others, get do lots of physical exercise and also mental exercise. Okay, that keeps you very active, and away from Alzheimer disease, at least for for many years.
Yeah, that's a very good take-home message, sir. And Dr. Anil is asking, does Alzheimer disease get transmitted? Like, is it communicable kind of from one person to the other?
Okay, very good question. In fact, I'm writing a book, and probably that book will be out in next few months. Okay, so it's covering all aspects, and in fact I have dedicated quite a bit of in fact a couple of chapters on these aspects. Okay, so I think he's talking about something called prion disease. Okay, and also, I don't know, vaguely you must have heard of mad cow disease. A few years ago, that has become a rampant in Europe. Okay, and this was a transmittable disease. And as of now, there is Alzheimer disease is at least not validated as a truly a transmittable disease, but there are enough indications. There are studies. For example, somebody has undergone a brain surgery. Okay, and then during that surgery, or for example, implantation of something, okay, the disease has been transmitted. Okay, in other words, other than these occasions, so it is not directly, there are no indications that direct communication or a transmission, but there is quite a bit of work is happening. There are some indications, but it is not like the prion disease yet. Okay, so that is the stage we are.
Okay, thank you, sir. I think, yeah. Thank you, Dr. Anil. And one more student, Abhi, is asking that along with memory loss, there are symptoms of confusion in the AD patients. How much is it related?
Yeah, very much. That's what I told you. So it's a thought, thinking, memory, okay, activity, everything, okay, learning. So in fact, confusion is part of all this. When I say cognition, I think it's part of everything. Okay, it literally, actually, that's what I told you. One slide I removed, not to scare you. Meaning, the person become a vegetative. Okay, the moment, let us say today, if somebody is diagnosed, after that you will have just few years, maybe 4 or 5 years of life ahead of him. And during that time, from the point of diagnosis, like a current diagnostic, after you will have a you may survive probably few years. and in that few years you will like kind of a vegetable. Literally you will, and then you will get irritated. They get irritated when they are having something, and then toward the end they cannot recognize themselves. Nothing. Literally they lose, they'll lose themselves. Yeah.
Okay. Yeah, it's really a very, what to say, scary disease to have.
Yeah, it is. Yeah.
And and as I don't see any more questions in the chat box here, but as you know, sir, we have gone live on YouTube as well. So I would like
Yes.
to take a feedback from Dr. Mrinalini. Dr. Mrinalini, do we have any question on YouTube live stream? Did the audience ask any question? Can you please enlighten us?
Not yet,
Okay.
Dr. Priyanka, not yet.
Okay. Thank you. And thank you so much, sir, for interacting so much. And the audience, I'm sure that they are very much enriched by all the things you told us about the Alzheimer's disease today. And
Thank you very much for having me, and I'm glad if they have benefited some little bit, from the.
Sure,
talk.
sir. We definitely have. And I would now request Dr. Ann Sandhya. Dr. Sandhya, are you there? Yeah, I would now request Dr. Ann Sandhya to present a formal vote of thanks to the speaker as well as to the audience here today. Over to you, Dr. Sandhya.
Good evening, everyone. It's my immense pleasure to give vote of thanks on the inauguration of BioRAM Forum for this academic year. On behalf of Department of Life Science, I would like to express my heartfelt thanks to our chief guest, Professor T. Govindraju, for taking his valuable time from a busy schedule and enlightening our students with knowledge. In particular, the concept of, from his talk, the concept of reactive oxygen species as targeting therapeutic target for neurodegenerative disease was thought-provoking for me. And he also well demonstrated the interface between the biology and the chemistry too. Thank you so much, sir, for enriching our knowledge with your talk. I also like to thank our chairperson, Professor M. Shivshankar, sir, for organizing this webinar, and also for being a constant motivator for all of us. Our thanks to Dr. Priyanka Sinha for hosting the session, Dr. Mrinalini Bia for introducing the BioRAM Forum or giving a brief introduction about the BioRAM Forum, and also for handling the technical session. Handling technically the session. Thanks to Dr. Anjali for introducing the speaker. Thanks to Saujanya, IBS student from 9th semester, for her wonderful Ganesha Vandane. And we wouldn't have accomplished this webinar without the cooperation of all the faculties of our department. Thank you each and everyone. And I'll not be fulfilling completing my duty if I don't thank our students. It's their enthusiasm and participation in this webinar made it really meaningful and fruitful. Thank you, dear students. Once again, I thank you each and everyone present here. Thank you.
And thank you so much, Dr. Sandhya, for that wonderful vote of thanks. And in his absence, because all of you know our chairman sir could not be here today because of some urgent and last moment work, he has personally extended his thanks to the speaker that he could not be here. He has apologized also. And also he has thanked all the audience members for this session. And thank you, sir, for coming.
Thank you, Priyanka. Thank you all.
Thank you.
Wonderful.
Okay. And dear all, this is not the end, but a new beginning, as all of you know. This day marks the inception of the BioRAM Forum activities for the academic year 2020 and 2021. So we are hoping to have a lot of activities, and I hope everyone enjoys these activities and participates in them. This is the message to our students. And today is also historical for our department because we went live on YouTube thanks to our team, which comprised of Mrinalini, Dr. Mrinalini, Dr. Anjali, and Ankita. And so we were live on YouTube, and our students could join us there also. And with these words, with the promise of a new beginning in this new year, which is coming up, we'll see you again. And thank you all. Thank you so much.
Okay. So thank you all. So I'll leave the room. Okay. So thank you.
Thank you.
Yeah.
Do I end the meeting?